Thank you for the introduction, and I would also like to thank the Belgian Society for Neuroscience for giving me the opportunity to present my work today, which is mainly focused on glutamate transporters in epilepsy. And today I will show you the results I got working on a model of viral-induced epilepsy. So let me introduce you to the topic, telling you that there are over 100 viruses which can induce encephalitis in humans. And viral encephalitis is often characterized by acute symptomatic seizures, which indeed occur in about 20% of patients. Beside the acute symptomatic phase, viral infections are considered one of the leading causes of the chronic condition of epilepsy itself. And indeed, there is an increased risk of developing late unprovoked seizures by 16 times following viral encephalitis, and this risk remains elevated up to 20 years later. Many potential mechanisms have been investigated in order to explain the development of epilepsy after viral infections, and nowadays a dynamic interplay between inflammation and neurotransmitters is likely to be important. Unfortunately, viral encephalitis and viral-induced epilepsy have been difficult to address experimentally because of the high mortality rate during the acute phase, but also because of the absent development of chronic epilepsy. Recently, a novel model of viral-induced epilepsy has been discovered by Libby et al. In 2008 after intracerebral infection of Taylor-Murine encephalomyelitis virus in black 6 mice. As you can see, after intracerebral injection, the virus spread bilaterally, mainly in limbic and the temporal regions, such as cortex and hippocampus and thalamic and septal nuclei. And after infection, about 75% of the mice develop tonic-clonic seizures. As you can see, at day 3 post-infection, the seizure activity starts, and we have a peak of seizures activity at day 5 post-infection, and at day 7 post-infection, the seizure activity stops, even though intermittent spiking can still be observed till day 14. At day 14, no viral antigen can be detected anymore in the brains of these mice. Therefore, these mice are able to clear the virus within 2 weeks. These mice were also subjected to some seizure threshold testing and corneal kindling in order to investigate if their seizure susceptibility was chronically alterated, and they saw a decreased seizure threshold and an increased susceptibility to kindling. And moreover, with chronic video EEG monitoring, they showed that 65% of these mice, developed also spontaneous seizures. Pathologically, this infection leads to different effects. First of all, there is neuronal loss that you can see, especially in the hippocampus, CA1 and CA2 pyramidal layers, while the dentate gyrus is almost intact. And this severe hippocampal damage corresponds to an enlargement of the lateral ventricles. Beside the neurodegeneration, they also observed gliosis. Microglia is activated, and there is also infiltration of macrophages, which occurs actually early after injection, because we see it already significantly increased after 5 days, and then it stays elevated also during the chronic phase. Also, astrocytes are activated, and this activation occurs between 7 and 14 days post-infection, so when the virus is being cleared and there are no more seizures, so it appears also involved in the clearance of the virus. Microglia and macrophages can release pro-inflammatory cytokines that were investigated, and we see in these graphs that some cytokines appear to be less relevant in this model, such as interleukin alpha and interleukin 1 beta, while TNF alpha, tumor necrosis factor alpha, was actually significantly increased only in mice which were having seizures 6 days post-infection. Interleukin 6 is also increased in this group, so mice that are having seizures, but also in the group of mice that is not having seizures, even though it was infected with the virus. These results were also confirmed by the fact that actually when these mice, the mice lacking of TNF receptor 1 and interleukin 6 knockout, only 10 and 15% of these mice developed acute seizures. These cytokines are known to be able to modulate glutamate homeostasis, acting on glutamate receptors and transporters. Glutamate is the main excitatory neurotransmitter in the brain, and its involvement in epilepsy is proved already, especially increased level of extracellular glutamate. And we know that the extracellular glutamate is mainly cleared by the excitatory amino acid transporters 1 and 2, which correspond to the rodents, GLAST, and GLT1, which are located on the astrocytes. We also know that the knockout of GLT1 leads to spontaneous seizures, so the involvement of these transporters has been already studied and described in epilepsy. We are actually more interested in another transporter, which is the glial cysteine glutamate antiporter, or system X-, that is also located on glial cells, microglia and astrocytes. This transporter is actually able to import cysteine, that then in the intracellular space is reduced to cysteine that is used for the synthesis of glutathione, and it's able to export glutamate. Of course, an increased activity and expression of this transporter could mean an increase in extracellular glutamate, also because it was shown that this transporter is the major source of extracellular glutamate in the hippocampus and striatum in the rodents. This induction has been shown to occur after many stimuli, such as oxidative stress and inflammation. Its involvement appeared very strong in epilepsy, and this is actually not surprising, but we know, for instance, that mice knockout of XCT, that is the specific subunit of system X-, are less susceptible to convulsants such as pilocarpine and cyanuric acid, and also that an increased activity of system X- has a convulsant effect. Moreover, in patients with temporal lobe epilepsy, mRNA and protein expression level of XCT were upregulated. Therefore, the aim of our work was to investigate the protein expression level of these transporters in an animal model, so in GLAST and GLT1, and of these transporters, so XCT. We did it by Western blotting using specific antibodies at 3 different time points, so 5 days post-infection during the acute phase, 14 days post-infection, and 2 months post-infection, so during the chronic phase. And we received cortical, hippocampal, and cerebellar samples. We pulled together the hippocampal samples because we received both contralateral and ipsilateral, and there was no significant difference. And we analyzed contralateral cortical samples. The cerebellar sample were used as controls, so the results are not shown. As you can see, at day 5 post-infection, we see in the cortex a significant increase in protein expression level in GLT1 and XCT, and we also see a trend toward an increased expression of GLAST in the group that was infected with the virus. In the hippocampus, we don't see significant changes, but again, we can see a trend, let's call it, toward an increased expression of GLT1 in the infected group. 2 weeks post-infection, so when the virus is cleared and there are no more acute seizures, we don't see any change in the hippocampus, but we could detect a significant decrease of GLT1 protein expression levels in the infected group. No changes were occurring in GLAST or XCT protein expression. During the chronic phase, we couldn't detect any significant change, but especially in the hippocampus. But in the cortex, we still could detect a trend towards an increased expression of GLAST and XCT. Therefore, we can summarize that we show that the glutamatergic system is involved also in this model of epilepsy. And actually, this is pretty obvious, especially in the acute phase when we see the increase in all the transporters in the cortex. We know that at this point, there is high expression of these pro-inflammatory cytokines. And we know that these pro-inflammatory cytokines have been shown to upregulate the expression of XCT in vitro. So we assume that that may also occur in vivo. This upregulation, as I said before, systemic C- is an important glutamate transporter that releases glutamate in the extracellular space. Therefore, these increases could be an attempt of the astrocyte to clear the increased level of extracellular glutamate. During the clearance phase, we don't see changes in the hippocampus, and we see a significant decrease in GLT1 expression. And in the chronic phase, even though these results are not significant, we still think they are pretty interesting, because here, if these increased protein expression level of XCT are actually correspond to an increased extracellular glutamate levels, these won't be compensated by the major glutamate uptake transporter, which is GLT1, that is responsible for 90% of the glutamate uptake. And therefore, this increase could actually lead to the spontaneous seizures we see in the chronic phase. Up to now, I know these results are purely descriptive, because we don't know that for sure that an increased expression means an increased activity. Therefore, further research is needed, and we are going to perform in vivo microdialysis experiments on black 6 mice infected with this virus in order to investigate their extracellular glutamate content, and also to in-depth investigate which is the role of systemic C- in this model. We are going to infect knockout mice deficient for XCT and compare their seizure phenotype and their epilepsy development with their wild-type littermates. I would like to thank my promoters, Isis Mulder and Anne Massy, Professor Karen Wilcox and J.C. Loven from the University of Utah for generating the model and sending us the sample, all my team, the XCT team, and all the Center for Neuroscience, and also all of you for the attention.
Okay, thank you, Giulia. Other questions? No questions? Okay, then we will move on to the next speaker.